Hello, everyone. Welcome to our MPRINT Hub webinar for February. Today we're going to be presenting an overview of the MPRINT Hub and some of our resources. You can see on the figure here our overview of the hub, including our cores here at Indiana and Ohio State University, and Copper, Logistics, Outreach, Real World Evidence, Knowledge Base, and Pharmacometrics and Clinical Trial Design Core. I'm Sara Quinney. I'm the PI of the MPRINT DMKRCC, along with Dr. Langley, who's also online and available for questions at the end of the webinar today. I'm also joined by our UCSD colleague, Dr. Shirley Tsunado, representing the UCSD Center of Excellence in Therapeutics. She'll be talking about their cores, including their pharmacometric and analytical chemistry core, microbiome analytics core, untargeted metabolomics core, and milk analytics core. And Prince Kannankeril from Vanderbilt University's Medical Center's Center of Excellence in Therapeutics will be talking about their work with their phenotyping core, placental biobank, and Precision Therapeutics Academy. We hope by the end of the webinar, you'll become familiar with the resources available through the MPRINT Hub and how to make use of those through our new and improved website and ability to access the resources that you may need. So the MPRINT Hub was established as a national resource to improve maternal and pediatric research outcomes by providing support at every step in the process. This is from helping to investigators identify research questions based on community needs, which is supported by our knowledge base and real world evidence core, as well as our Copper Biobank support services, which help you investigators identify gaps in knowledge and available resources to help with your research. Our pharmacometrics and clinical trial design core, as well as with experts from a variety from across MPRINT and outside collaborators can then help you support study design and strategic sampling. We encourage best practice protocols and custom support for study design to maximize the outcomes. And then following, studies, we can support the analysis through, especially through the UCSD cores for metabolomics and pharmacometrics and analytical chemistry core, as well as the integration of data and biostatistic and pharmacometric analysis through some of our other cores. And then we have, through our website, our webinars, meetings, and other resources, ability to outreach to make our research more shareable. So I'm going to start with describing a little bit about what the Data Model Knowledge Research Coordinating Center has available to support research in maternal and pediatric therapeutics. And I'll start with the knowledge base. This is led by Dr. Langley from Ohio State University. And it is a curated database of data from PubMed and other resources focused on maternal and pediatric pharmacology. So there are two versions available. The gold version is a manually curated database, which is continuously being updated. And our primary focus at this time is on drugs from the BPCA and PREGLAC prioritization lists. And then continuing to update in the future to add pharmacometric models, more pharmacodynamic data, and potentially some in vitro data as well relating to ADMET and relevant pathways or proteins. The silver version is a larger collection of abstracts that were selected using a natural language processing tool, which are classified as either pharmacokinetic, pharmacoepidemiology, or clinical trial papers. And again, this is continuously being refined. Here you see some information on statistics on the abstracts and publications and data we have available in both the gold and silver knowledge base. You can see we used a BioBERT model for our silver natural language processing model where we had over 4,000 labeled abstracts that were used for training and validation of the model. From that, we were able to identify from PubMed over 750,000 relevant abstracts, and currently have about 3,800 of those manually curated into our GOLD database. You can see on the right some statistics of the population. Obviously, we have more pediatric than maternal studies. Most of the data collected in abstract form is from pharmacoepidemiology studies. And then we have it broken down by subpopulation here in the bottom. In the GOLD knowledge base, we've looked at about 164 drugs in total here, and you can see we have 139 we have pharmacokinetic data on, and 164 we've looked at from a pharmacoepidemiology and clinical trial standpoint. We can see again that there's more data for pediatric populations, but a good amount in our database on maternal populations as well. And you can see in the chart here some of the PK and outcome parameters that we are assessing in these papers. You can access both the gold and silver knowledge base through our MPRINT website, which we will show in a little bit through the resources page. And this is just a screenshot of the knowledge base, the gold version, where you can search by drug name and then filter the data based on population and study type. For the silver knowledge base, it has a similar portal that you can access through the website. And again, search by drug. Also, we're allowed capable of searching by a disease or a physiologic state name. And then in both, you can download the data once you select the information you would like. We also have a project, the Collaborative Online Perinatal and Pediatric Repository, or our COPPER repository. And this is a website that collates biobanks from across the country and across the world. We currently have 16 biobanks represented that contain samples from maternal and or pediatric populations. And most of these studies include hundreds to thousands of participants and over 500 samples available. So this is a screenshot of the homepage for the COPPER website, again available through mprint.org. We have ability for you to search by the category of biospecimen you are interested in, as well as by a number of fields, including age, condition, participants, demographics, and so forth. We also have ability for you to submit additional information to our biobank here, and we are continuously growing this. So the goal of this is to provide linkage to biobank specimens for investigators who may be searching for samples to analyze. Once you go into this, you are connected to an email for each of the biobanks that you can then contact for more information if they have specific samples of interest to you. The Pharmacometric and Clinical Trial Design Core is co-led by Dr. Robert Bees from University at Buffalo and myself. And we work in partnership with a number of investigators and organizations, including the MFMU and Pediatric Trial Network and Impact, along with investigators from multiple universities, to provide resources and support for pharmacometric modeling. We have expertise in monoclonal antibodies, maternal and pediatric physiologically based pharmacokinetic modeling, population pharmacokinetic and pharmacodynamic modeling, as well as support for study design. We also bring together expertise. So if we have had meetings with a number of pharmacometricians from and clinical pharmacologists from across the world through ACOP and other meetings to help further expand and bring together that expertise. We support the Clinical Pharmacology Training Network, participating in their meetings and webinars, as well as providing mentorship for clinical pharmacology trainees. This coming summer, we will be supporting a workshop on population pharmacokinetic modeling in conjunction with the CPTN annual meeting. The Indiana CTSI has an annual modeling and simulation symposium. You can see here the upcoming information. Last year's symposium was co-sponsored by MPRINT and had a focus on maternal pediatric therapeutics. This year's theme is in novel modalities and novel therapeutics, but also will be of interest, which, while not specifically maternal pediatric related, will be of interest to those in the field as well. And we'll also provide trainee opportunities for networking with individuals from other academic centers as well as from industry. We also are active in supporting publications. For instance, we supported a special interest issue of pharmaceutics on physiologically based pharmacokinetic modeling in pregnancy and lactation, and have helped co-edit other special issues of journals relating to pregnancy, lactation, and pediatrics. And finally, we present at annual meetings at ACOP. We've previously had discussion sessions bringing together those experts from across the world, as well as hosting symposia to focus on maternal pediatric pharmacometric aspects. I'm going to now turn to our real world evidence core. This is co-led by Dr. Chris Bartlett and Sean Granis from Nationwide Children's Hospital and Indiana University Regenstrief Institute. Also Anita Mendoko from Cincinnati Children's Hospital is highly involved. And the goal of this is really to connect maternal pediatric real world evidence resources from across institutions. So we have ability to connect with resources from across Indiana through the Indiana Network for Patient Care run out of Regenstrief Institute, which has EHR data from over 70% of Indiana, along with data from Ohio State University and Nationwide Children's and Cincinnati Children's, which covers quite a large portion of Ohio. The Real World Evidence Core is working to establish linkage models to connect maternal and child data across EHR records, and as well as using natural language processing methods to identify social determinants of health that can then be integrated into analyses. Both Nationwide and Indiana University have biobank samples that can be linked to the EHR data. And finally, they're working to further define pregnancy and lactation phenotypes. With that, I will turn it over to Dr. Tsunado to talk about resources from UC San Diego.
Thanks so much, Sarah. I'm really happy to be representing the UC San Diego Center of Excellence in Therapeutics for MPRINT. We consist of all of us here. The title of sort of our program is Optimization of Antibiotics in Mothers and Their Breastfed Infants Using Pharmacomicrobiomic and Metabolomic Analyses. Our administrative PI is Dr. Audrey Tremoulet, and our co-admin PI is Tina Chambers. I, together with Peter Dorenstein, run our data science project. We also have a basic science project run by Victor Nizet and George Liu. We have a milk analytics core that's headed up by Lars Bode, and then we have a pharmacometrics and analytical chemistry core headed up by Jeremiah Momper and Brookie Best. Next slide, please. So At our UCSD Center of Excellence, we interface all three projects together. We currently have a clinical project that just finished recruiting. We've collected all of the samples for this clinical project that I'm going to tell you about in a minute. And we're currently processing them in our untargeted metabolomics pipeline. Concurrently, we have several ongoing studies with our basic science core using animal models to investigate some of the questions and the mechanisms of the questions that we're interested in with our clinical project. And this is all supported by our milk analytics core and our pharmacometric and analytical chemistry core. Next slide. So at UCSD, we're very interested in the mother-infant dyad, and from pregnancy to early life, and the intersection between the microbiome, the metabolome, and human milk oligosaccharides, which sort of are thought to be food for the microbiome. And then on top of that, we're very interested in environmental influences on this triad. For example, exogenous drugs, food, and personal care products. And how does this influence the development of the microbiome, the infant metabolome, and the contribution of HMOs through breastfeeding on this cycle, and how this affects the immune system and the potential health of the baby. Next slide. One of our foundational cores here at UCSD is the Human Milk Biorepository that was established by Tina Chambers many years ago. And she has lots of collaborations with various different networks. If you want to go to this site, please feel free to use this web link. And this will take you to this web page where if you would like to do a project using human milk, you can apply to obtain some human milk samples. Currently, there are over 100,000 human milk aliquots that have been donated by over 3,000 individual women. Next slide. And so Tina has also led our clinical study where we enrolled, this started back in 2020. So we enrolled 80 mother and infant dyads, and we were very interested, again, in the question of when a mother is prescribed antibiotics for whatever reason, and she's breastfeeding. Obviously, we have a lot of information about the drug in the breast milk and, calculation of dose to the infant. But what we're really interested in is how do these antibiotics impact potentially the developing microbiome of the infant and subsequently the metabolome of the infant. And so we set up a clinical study to try to answer that question. And we have sampled fecal samples, skin samples, breast milk samples, as well as environmental samples of the baby and mother to answer a host of questions. And so we're currently analyzing all of these samples. We've collected them all now. Tina's team has successfully completed enrollment. We are determining the pharmacokinetics of 4 of the commonly used antibiotics in the milk. We're trying to answer the question of what is the impact of antibiotics on milk composition and HMO profile. And then what is the impact of antibiotics via breastfeeding on infant growth and development? So again, Tina's team has collected a host of metadata and is following the infants over time to try to answer some of these questions. Next slide. Lars Bode runs our milk analytics core, and he has a host of techniques and technologies to enable anybody to investigate the composition of milk and human milk. So his vision for this milk analytics core is to become a comprehensive core with knowledge, skills, and technology that can serve as a resource to this and other MPRINT CETs, as well as the research community at large. Next slide. And his team has various technologies and assays to be able to answer a host of questions with respect to the composition of human milk. And as common as human milk is, and as a, you know, golden liquid gold, as we call it, to human development, there is not that much known about human milk. In fact, you know, whether or not what the microbes in the human milk are doing, I think for a long time thought that milk was sterile. Well, it's not sterile, and there is actually a microbiome associated with human milk that we hope to study as well. So his group can look at the macronutrients of milk in addition to the micronutrients of milk. He can also characterize the milk for human oligosaccharides, proteins and peptides, fats and proteases. Next slide. And Lars has also pioneered our Human Milk Institute at UC San Diego. We are in the third year, I believe, of having an annual symposium devoted to human milk. Lars tells me it's the only institute worldwide devoted to the study of human milk. And this coming March, we are doing a deep dive MPRINT session hosted by Tina Chambers to explore some of the research that is going on in human milk and that relates to maternal and pediatric therapeutics. Next slide. And so this is going to be held here in La Jolla, March 4th through 6th, and I believe it's already sold out. So if you want to attend, please get on the wait list. But we'd love to see you at the beautiful Scripps Seaside Forum in La Jolla for a 3-day symposium. Next slide. Our basic science core has been very busy, again, looking at the impact of antibiotics on the microbiome of an infant and how this mechanistically may impact the developing immune system. And also at the same time to look at human milk or maternal milk oligosaccharides, not human, because this is a mouse model, and how these milk oligosaccharides might contribute to a more diverse microbiota, which then might recover some of the immune competency that can be compromised by antibiotics. Next slide. So Doctors Nizay and Liu have been conducting several studies in an animal model, and they are very interested in looking at exposure to ampicillin in particular, because it's a very common antibiotic given for group B strep prophylaxis to mothers, and to look at the impact on the immune system by looking at immunization efficacy. Next slide. And so this is just preliminary data where they've shown giving either a hepatitis B vaccine or a Prevnar vaccine after the baby is exposed to ampicillin via breast milk, and does this impact the response to the vaccine? And what they did show was that with the Prevnar vaccine in particular, ampicillin knocked out that IgG response. Next slide. There's several other projects going on in the basic science space, and this was a support pool grant to the group looking at the impact of maternal milk cytokines on the infant microbiome and the susceptibility to enterocolitis. Next slide. And another support pool funding grant that was given to Dr. Pia Panaraj to look at the impact of maternal milk IgA binding to fecal bacteria on an oral rotavirus vaccine response in infants. Next slide. For our pharmacometrics and analytical chemistry core, this is run by Jeremiah Mumper and Brookie Best. We have the ability to run virtually any targeted quantitation assay for drugs, for proteins. And our clinical project, our data science project, and our basic science project all converge on utilizing the expertise of this core. Next slide. And there's the ability of this core to also provide innovative PK modeling and PBPK modeling as well of samples that they receive. Next slide. And here are just some example projects that they're involved in right now. And what you'll notice is that around the country, various investigators have partnered with our pharmacometrics and analytical chemistry core to take advantage of some of their bioanalytical expertise. So Cincinnati Children's, University of Utah, and several other networks as well have utilized the services of the core. Next slide. So together with Peter Dorenstein, I run our metabolomics and pharmacomicrobiomics program, where we're really trying to leverage analytical and computational tools to answer important questions in the maternal pediatric clinical pharmacology space. And we're particularly interested in host microbial metabolism. Peter's group has discovered many new bile acids, which we believe are important communicators between the host and the microbes. His group has developed many computational and analytical tools. And by the way, these are all publicly available to any of you to learn. And there's also many tutorials available if you're interested in learning how to use these tools to answer some of your research questions. We have created a drug library to annotate more drugs and drug metabolites from mass spectrometry data. And Peter is also developing a microbial metabolite knowledge base, and this will hopefully be a powerful tool to help us understand some of the function of the microbiome metabolites. Next slide. So this is a study that Nina Zhao, one of our postdocs in the group, has. It's a paper that is currently under review. And she basically leveraged publicly available data from various sources and has been able to now provide an annotation source of drugs and drug metabolites based on mass spectrometry data. Next slide. And I'm very excited about this drug library because this will enable us to annotate more drugs and drug metabolites from all the samples that we are analyzing, and any of your samples. If you have, a matrix of interest, be it skin, breast milk, fecal samples, plasma samples, we will have the ability now to annotate more drugs and drug metabolites. And notably, my fellow Quinney has collaborated with Nina on this paper to provide a metadata connection to these mass spectrometry annotations so that there's some context for what we have annotated. Next slide. And now I'm just wanted to tell you about a developing story that we are involved in, and this is unpublished data. This is just another example of some of the research that we have the capability of doing, and particularly of combining the microbiome and the metabolome analysis. So this is a study that we are fortunate to be collaborating with the Gates Foundation on. In a mother-baby dyad longitudinal study in Bangladesh. And so these were mother babies that were recruited to the study, and a longitudinal sampling was done of feces, tongue samples, skin samples, human milk samples, vaginal fluid, and plasma. And we have now analyzed this data both for the microbiome as well as the metabolome. And I will show you a little bit of the data, but just to sort of help you understand, what's the importance of this study. And it's really an observational study in a non-Western population, which is rare. Most of the microbiome and metabolome data that we have, particularly in the mother-infant population, is from Western populations. So this is particularly interesting that we were able to collaborate and be able to analyze these samples. Next slide. And so I'm just showing you the metabolome data. And interestingly, what we've shown, and this is sort of a trajectory of what the human milk metabolome looks like over time and what might be influencing the development of the different metabolites that are seen in human milk. And so what we found was that delivery mode, so whether or not the mother delivered via C-section or vaginal, HMO secretor status, and actually exposure to filtered water impacted the development of the milk metabolome. And so again, this paper is in development, and hopefully that will be out there in the scientific community for you to review. Next slide. So finally, I just wanted to end with another sort of schematic of some available analytical tools and databases that are available to you. This is all, we very much believe at UC San Diego in public availability of our tools so that we can share knowledge within the scientific community. And so we are developing a microbial metabolite database that harnesses publicly available data from around the country so that we will be able to start to annotate all of these metabolites that are produced by microbes. And this will be in concert with phenotypic data and metadata that we can, disease-based data, for example, whether or not the sample is in an animal or a human. And we will start to be able to make connections with your help to understand what all of these microbes are doing in the microbiome and understand this through analysis of the microbial metabolites. Next slide. So I just wanted to conclude by saying we're very open to collaboration. I hope I've outlined to you some of the resources that we have at UC San Diego. If you're interested in collaborating with us, please feel free to reach out to me or to Sarah, and I will connect you with the appropriate people at UC San Diego. Thanks so much.
All right, so I'm up next. My name is Prince Kannankeril, and I'm the contact PI of the Vanderbilt Center, which we call Vice MPRINT. Digna Velez-Edwards is the co-PI along with me, and this slide just gives sort of a high-level overview of the different projects and cores that we have going on. Project 1, which is a clinical project focused on pharmacogenomics. Project 2, more of a data science project focused on maternal opioid use disorder, led by Stephen Patrick. I'm not going to go into a lot of details about either one of the projects specifically. The administrative core, led by myself and Digna, we sort of supervise the Precision Therapeutics Academy, as well as the support pool projects and some of the newly funded initiatives that were supported by administrative supplements. And I'm actually going to spend most of the time talking about the work that's being done by our phenotyping core. And this is led by Wei-Chi Wei. And so you can go to the next slide there. So the phenotyping support core, they really support everything we do, both the projects, all of our trainees. They have 3 primary aims. One is to create and validate phenotyping algorithms. Second aim is to assist with replication of findings from other sites. And then third is to develop and share tools for MPRINT research. And predominantly what they do is they work with electronic health records data and large data sets, particularly those tied to large biobanks. And they use tools like natural language processing, machine learning, and so on. And they develop these algorithms which they share widely on PKB. So if you go to the next slide, this was just a screenshot from the phenotype knowledge base, which again, any of you can access. And if you just look at the most recent phenotypes there on the right, you'll see the third one down is opioid exposed infants. So that was a phenotype that Wei-Chi's core actually developed for Project 2 for Stephen Patrick and his team. It's a little more straightforward to find in the EHR patients with a specific diagnosis like neonatal opioid withdrawal syndrome, but finding infants exposed to opioids who don't necessarily have a diagnosis is a little bit more tricky. So you can go to the next slide. So this paper's been published, and again on VKB, you can if you click through it, you'll see sort of the details of how they did it. And then actually, if you click to the next slide, you can actually get all the specific information you need to deploy this phenotype in your own data set if you like. And you can see it uses some natural language processing as well as structured data, including CPT and ICD-9 codes. So, and again, this is all made available for anyone to use, both on GitHub and at VKB. If you can go to the next slide now. And so one of the things that I did want to spend a little time explaining is the development of pediatric fee codes. And as I was doing this, I realized not everybody lives and breathes fee codes, so I might have to give a little bit of background on what that means. So you can go to the next slide. Back in 2010, Josh Denny, who used to be here at Vanderbilt, now runs the All of Us program at NIH. He came up with the idea of a phenome-wide association study, or a PheWAS study. So most of you understand what a genome-wide association study is. You've got a disease like diabetes, and you've got cases and controls, and then you can look across the entire genome to identify novel, gene disease associations. And so Josh realized, you could do the opposite of that. If you've got a large biobank linked to electronic health records data, you could take a gene or variant of interest and then run for associations across all phenotypes. And so hence the name phenotype-wide association study. So when he did that, it was just lining up ICD-9 codes. And so this was just an example of a variant that was known to associate with multiple sclerosis, just showing that, yes, you can identify patients with multiple sclerosis who have that gene of interest using this phenome-wide association study. And then the little graph on the bottom left there, that just shows if you PubMed phenome-wide association studies, you can see the increase in those studies that have been done since he described it in 2010. So last year there were 157 studies in PubMed that at least use that term PheWAS. So all the cool kids are doing it, right? So you can go ahead to the next slide. And so Wei Chi, who again runs our phenotyping support core, he published the concept of fee codes. So ICD-9 codes, of course, are billing codes, and they're not what what clinicians really would use or what researchers really use when doing precise phenotyping. And so Wei Chi and his team sort of went through the process of refining those codes into more clinically and research relevant fee codes. And so this was a paper just showing that you could use that fee code sort of structure, and that they perform better than either ICD-9 codes or CCS, which is something that was developed by AHRQ. So fee codes are really what people are using for these phenome-wide association studies. And so now you can go to the next slide. And this is a project that was done by Monica Grabowska, and this really would not exist without MPRINT support. So she really led this project developing PEDS fee codes, or pediatric fee codes. So if you go to the next slide. So she did this by basically taking the original fee codes and then sort of doing a parallel process. So she looked at fee codes that had higher prevalence among pediatric patients compared to adult patients. And for many of those, we realized we needed to make new fee codes. So, if you've got a fee code that just says, congenital abnormalities of the circulatory system, that may be fine, for an adult researcher. But, if you're a pediatric cardiologist like me, you want a lot more precision. So we created a lot more specific fee codes for the pediatric population. And then advance the slide, and it's just going to show the other side. So some fee codes were very rare in the pediatric population compared to the adult population. So occasionally we want to keep those. They may just be, diseases that are just less common in kids, but many times we would remove them. So, prostate cancer, Alzheimer's disease, those are diseases that really aren't relevant to the pediatric populations. So we remove many of those. So you can advance the slide. So, and this just shows sort of on the left some of the common diagnoses that are more prevalent in pediatrics on the top and more prevalent in adults on the bottom. So you can see otitis media, less than 2% of adults have that, but more than 10% of kids have that. That makes perfect sense. And then if you look at GERD, it's more prevalent in adults, but still fairly common in pediatrics. So we want to keep that one in. And so on the right, you can see the Venn diagram. There are over 1,600 codes that remain the same, but we eliminated 173 fee codes for the pediatric population, and we actually added 358 new codes for the peds group. And it's actually more complicated than that with sort of the stem and leaf terminology. We don't need to get into that. If you care about it, you can read the paper, but you can advance the slide, which just shows that the peds fee codes for pediatric diseases, of course, performs better than the adult fee codes do. So one example here, a known association with Hirschsprung's disease, you can see that when you use the pediatric fee codes. If you use the standard fee codes, it's sort of buried under congenital anomalies of the intestine, because that, of course, includes many other congenital anomalies that are not specific to Hirschsprung's disease. So it's not really as useful for the pediatric population. You can advance the slide. So again, we expect these pediatric fee codes will facilitate much better large-scale phenotype analysis in pediatric cohorts and in populations. And again, this is all readily available on Wei Chi's lab website. He's got sort of the details with PEDS, PWAS, but it's also available at feecode.org. So you can look there if this is something that you're interested in. So just I'm going to just quickly touch base on another sort of our initiative. So the Precision Therapeutics Academy, led by myself and Digna, this is an effort where we train and mentor junior investigators who are doing precision therapeutics in the maternal and pediatric space. So we create an individualized training program for each of them. Really, the hallmark is a mentored research project that they complete during their 1 or 2 years. We sort of make it flexible for the individual. And all of them, of course, are expected to participate in MPRINT webinars like this one, as well as some of the shared resources that the MPRINT Hub puts on, as well as many of the relevant seminars that we have locally here. You can advance the slide. And then I think the last thing I'm going to touch on is as part of the PRI-3 initiative here, we've developed a placenta biobank, which I think is good, a little described on the next slide, so you can advance that. So Project Placenta, and this is something that was expanded with use of an admin supplement from MPRINT. So this is a biobank that takes the placenta, which of course is usually just discarded after birth, and we can extract both maternal tissue and fetal tissue from this one source. So it's a rich source of tissue that can inform both the maternal and the fetal genome. And so Digna is currently doing a project it's focused on gestational diabetes and the use of different medication exposures during pregnancy and how it affects both maternal outcomes and pediatric outcomes. But obviously, there are many more opportunities that can be explored through the Placenta Biobank. And so if you've got any interest in that, contact us and we can get you in touch with Digna and her team. So I believe that's my last slide, so you can advance that. Yes, and now I'm going to turn it back over to Sarah, who is going to talk about opportunity pool funding.
So, thanks, Prince and Shirley. Great opportunities and resources available through both of your CETs. Yeah, so I was going to talk a little bit about the opportunity pool funding awards. We're distributing our awards this year. We are going, as compared to the past, we are going to have still 2 awards for trainees, so fellows or postdoctoral researchers or even residents and graduate students can receive up to $25,000 in total costs to support your project. That RFA will be available through our website in the next shortly, if it's not already there. The rest of the funds we have that we've currently used for RFAs, we are going to be using for improving the resources and developing collaborations with the existing cores of MPRINT. And so For instance, if you're interested in collaborating with our real world evidence core, our knowledge base to obtain data, we may be able to provide some support for those projects. Or for instance, with pharmacometrics clinical trial design core, as well as the phenotyping cores and the cores at UCSD, we may be able to support some resources through there. Proposals will be accepted from clinical or basic science investigators, and largely they're going to be we're going to focus funding on things that are meeting emergent needs and leveraging novel technologies in maternal and pediatric therapeutics. And as I've mentioned, we have a new website that has recently launched. You can still find it at the same place at MPRINT.org. This is the face page. Emily Kinney has done a great job in putting this together, and hopefully it's a little bit more user-friendly for you, and you're able to identify the resources that we have available. You can see at the top of the screen, there's a number of headings that make available the various resources. You can find information on our events, as well as on our opportunity and support pool funding programs. Go down. For instance, if you click on resources, it will take you to another page that will link you to all of our individual research cores. And then you can click on those cores and it will tell you a little bit about what we do and provide contact information for you to contact each center, as well as some of the resources that are made available through those centers. We also encourage you to reach out. We have an email list available that we send out quarterly emails through the listserv, but we also with newsletters, but we also send additional information relevant to maternal or pediatric therapeutics. You can reach us by email if you have questions about any of our resources, info@mprint.org, and also follow us on social media. On X, we are Imprint Hub, and on LinkedIn, also Imprint Hub there. And then we also have a YouTube channel where this and other videos are posted for your information. With that, I will thank you for attending today and open the floor for questions. Please let us know if you have questions for any of our centers, or we have some of our other colleagues on the line that may be able to answer your questions. I guess I can start. I'm gonna reach out to see Chris Bartlett is online. If Chris, if you can come on. So, I talked a little bit about the real world evidence core, but can you give some examples of the work that you're doing through there?
Sure. So we've got a number of things going on. We have the sort of computable phenotype realm where we're working on record linkages across institutions, finding moms and babies and continuity of care, computable phenotypes like lactation, pregnancy, things like that. The other side of that is working on projects where people are actually sort of doing something with the data itself. I think one good example was we pulled drugs that were being prescribed in various age groups, and then work with Long's group to get the intersection of, so we can say what drugs are being used, and then Long could say exactly how many publications were there for each type of drug for each interval that we're interested in, pregnancy versus lactation, and look at things like that. So just one example, or several examples of things that we're doing, but always interested in additional projects being pitched to us so that we can help support them.
Shirley, I see a question in the chat for you for the animal models for lactation studies where you're using mice. Is that an appropriate model for lactation?
We believe so. We are using mice as our animal model for lactation. To be transparent, we have been having difficulties with survival of some of these babies. So we had quite a bit of, cannibalism by the mothers. And again, we're not quite sure. So it is an appropriate model, we believe, for also studying, the immune system effects on the babies, but it's not without its challenges. I hope that answered your question.
I think I was just gonna comment on that. I mean, the data you showed showing how like access to filtered water changed the microbiome, it just shows how complex it is. I mean, obviously there's gonna be huge differences that you can observe between the mouse, based on, how they're being fed and all that stuff. But it's just, every time I hear you guys talk about this, it amazes me how much there is to learn about human milk and how much we don't know. It's like you said, it's been around forever, and there's, it's so ubiquitous, yet we know so little about it. It's really fascinating, all the work you're doing.
Yes, thanks, Prince. And as you know, we're trying to get at it from multiple angles. So thus the mouse model, which, is not without its difficulties for interpretation and challenges with handling the mice, but we are able to do things obviously with the mouse that we aren't with the humans. So it's the pros and the cons.
So looks like your mom made another comment about the pH of the milk in mice being different, so it may impact partitioning of the drugs into milk as well. So
yeah, that's interesting. And I will tell you that the work that we're doing in looking at the impact of drugs on the metabolome and the microbiome, we are doing that in humans. So we're not, and also some of the work that Jeremiah Momper's group is doing as well with looking at the pharmacokinetics of the drugs as they go through the milk, that is also done in humans. So to minimize the trans species differences in the breast milk. So most of the work that we're doing in the animal model has to do with more mechanistic changes in the immune system, and using the breastfeeding as a vehicle for getting the antibiotic into the infant. I hope that helps.
other questions or comments? If not, I've just put the address for our website in the chat. Please check it out. Feel free to reach out to us at info@mprint.org or to any of the speakers here today if you have questions or would like support from MPRINT. We really enjoy meeting with people and discussing ideas and projects and trying to help troubleshoot or connect you with the right resources. And even if that's not within MPRINT, we utilize our network to help identify those resources as well. So not seeing anything else in the chat. So thank everyone for attending and your attention today, and we will see you next month.
Thank you.